Thank you for inviting me here. So I'm glad to give this talk. This topic is about an overview of the challenges with PPCPS in China and advanced treatment techniques. So the topic will, firstly, I will give a general introduction about the challenges of PPCPS in China, including the production, consumption, and occurrence of PPCPS in the environment of China. And the second part and the third part will relate to our former research work about the typical PPCPS treatment by some advanced techniques, for example, UV Paxlovid and other techniques. Now, let's move on to the first part. So, PPCP are widely used by humans and animals. So the main sources of the PPCPS are hospitals, farms, and wastewater treatment plants. So until now, we know that many kinds of PPCPS cannot be removed by the novel treatment techniques. So just releasing a lot of compounds into the environment, which is the main source. Many studies have focused on the occurrence and behavior of PPCPS in environment. So the same in China. So we have many studies on how to find the typical PPCPS in the environment. So there are several representative PPCPS, antibiotics and estrogen, and anti-inflammatory, analgesic, and mask present in cosmetics and detergent. Among them, antibiotics are a very severe problem in China. So China is the biggest or the largest production and consumption of antibiotics. According to results conducted recently, about 75% of penicillin and 80% of cephalosporins and 90% of streptomycin were produced in China. The total antibiotics produced in China is about 248,000 tons. Only 1/3 of them exported to other countries. The rest are used in China. And for the antibiotics used in China, so human use accounted for about 48%. From this table, we can see that the USA only use about 11% of the antibiotics as that in China. So this is a very big challenge for Chinese. Among these 162,000 tons of antibiotics. Human use are about 48. The antibiotic abuse in China is very urgent or very emergent problem. So who should be responsible for this situation? I think first is the hospital, then the consumers, last is the livestock. The hospital or the doctors, usually people, I think there are a lot argue about the doctors. The doctors will prescribe a lot of antibiotics to their patients for some financial incentives. And also, I think there's a big difference about the doctors in China and in the USA or in other countries. I have an example. So one doctor in China will see about more than 100 of patients, which is several times more than the USA. So they have a very short time to make a decision about the patient's treatment. So they only could use the very quick and effective method to treat their parents. And for the consumers, because they can easily get the antibiotics from the pharmacies, so, and also they have the experience from the doctors, many kind of illness like the cold, like cough, sore throat, can easily be treated by antibiotics, so they will directly buy the antibiotics from the pharmacy. So without the scientific guidelines, consumers can use a lot of antibiotics. So the third reason is the livestock. The abundant use of antibiotics worldwide is livestock, especially in China. So China every year will raise about 12 billion of chickens and 1.2 billion of pigs, and about several hundred millions of cattle and sheep. That's a great pressure to the environment, because the farmers will use a lot of antibiotics to feed the animals, to keep the animals alive, and to make the animals maybe low fat and overfed, something like that. So this is the problem. The consumption data of antibiotics in different countries from in different years show that China, the defined daily doses for 1,000 inhabitants per day is about 6 times higher than that in the USA, in the Canada, in Europe. So that's very big, very large contamination for China. So after use, many of the antibiotics will be excreted and changed or as metabolites, where the feces and the urine just continually releasing into the environment. From this table, we can see the target antibiotics. The average excretion rate is about 80%, and for the anti-inflammatory, so more than 90% of the drugs will be excreted into the environment. Some compounds will be excreted completely. There are many studies about the antibiotic emission in the environment in China. From this map, we can see the total emission, environmental emission of the antibiotics for each river basin in China. So the red and the yellow river means the emission are very huge. And also there's another map, the emission density about each river basin of the antibiotics in China. So from this map, we can see the Pearl River Basin located in the south of China showed the highest emission density, followed by the Hehe Basin, and Hehe is in the north of China, and followed by the Tai Lake and the Qiantang River located in the east of China. So in this area, the mega cities located in this area, Beijing, Shanghai, Hangzhou, and Shenzhen. So in general, we can see the average emission density of this of the east and the south of China are 6 times higher than the west area. So we can draw a line in this map. This line is very, very useful. On the left of the line is undeveloped, low population, and on the right of the line is economically developed with a large population, and this line will reflect how huge human activities on the antibiotics distribution. So the same distribution characters are also for the other personal care products and other drugs. Just now I mentioned the mega cities. Mega cities located in Zhujiang Delta, Hangzhou, and Shenzhen city. So in Yangtze Delta is Shanghai, and Jingjiangji region is Beijing. So from the concentration of the wastewater effluent or effluent, we can see that in this area, the concentration of the antibiotics is very significant. In the following, I would like to introduce our former research work about PPCPS detection in Shanghai area. Shanghai is located in the Yangtze River Basin, so it's a very economically developed area. Shanghai has a population of more than 20 million. So in this area, we can see, we want to know how the occurrence of the antibiotics or other drugs in the environment. We choose 8 frequently used fluoroquinolone in our study. So the sampling site is in some typical sewage treatment plants and also in the island surface water. So some of the fluoroquinolones are used for human, and some of them are used for animals. So from this results, we can see almost, except the SAR compounds, most of the fluoroquinolones will be found in the effluent of the sewage treatment plants. And the 2 human-use antibiotics were found in every sewage treatment plants. The other animal-use compounds like DANO and ENRO and DIF were detected in the surface water. So from the overall removal of the fluoroquinolones in sewage water treatment plants can be shown very efficient, but not complete. That just let treatment of the fluoroquinolones in the receiving waters. So next we do some research about carbamazepine. Carbamazepine is an important antiepileptic drug. So carbamazepine in our study was the sampling size of our study is in Shanghai and in Chongming Island. So along the Huangpu River, we chose some sewage treatment plants and some surface river to detect the carbamazepine. So the results show that carbamazepine was detected in Suzhou River. Suzhou River is the mother river of or the local river of Shanghai. So Suzhou River has the highest concentration of carbamazepine. So, that's a big challenge for Shanghai because it's the main river for Shanghai. So another river is a river in Tongji University. That's our campus university. So we can see the carbamazepine, the concentration of carbamazepine is about 190 nanogram per liter. So I think this is a challenge for our department because we are working in this university. The carbamazepine in the sewage treatment plants, except one sewage treatment plant, we can't find the carbamazepine. The rest, effluent, we also find the carbamazepine. And it's very interesting that the effluent of the sewage treatment plant, the concentration of carbamazepine is higher than the influent. So some other studies also have these similar results, but we don't know why, because my student graduated and this work stops. So, but we do know that carbamazepine could not be removed or eliminated from the secondary treatment plants. The following, we do some research about acidic pharmaceuticals. Acidic pharmaceuticals is very kind of drugs for the human health care. According to some studies, acidic pharmaceuticals are poorly absorbed after intake. Most of the compounds, about 40 to 98% of the injected compounds will releasing to the environment. So in our study, we choose six kind of acidic pharmaceuticals. We didn't do research on the last one, so we have 4 anti-inflammatory and 2 used as the medicine to regulate blood rapid. So this is the sampling size. We have 3 samples from the main area of Shanghai along the Huangpu River, and 4 samples from Chongming Island. Chongming Island is normally mainly depend on the agriculture, no industry. So the results show that all the compounds were found in the 17 sites. The highest concentration of the target acidic pharmaceuticals were 224.4 nanogram per liter. So this is the situation of acidic pharmaceuticals occurrence in Shanghai, in the surface water or in the effluent of the sewage wastewater. To summarize, China is the largest PPCPS production and consumption country in the world. So the PPCPS is a very severe problem for China. So the secondary drugs and antibiotics would be excreted into the environment and can be detected widely. So this is a big challenge for not only wastewater treatment, but also for drinking water treatment. And novel techniques should be developed to remove typical PPCPS, especially in wastewater treatment plant, because wastewater treatment plant is the last barrier or the last defense for aquatic environment protection. So that in the following, I will give the introduction about the techniques, the advanced techniques we use to treat a type of antibiotics, penicillins. Penicillins is most frequently used as the least toxic antibiotics. So penicillins, for example, accounted for about 44% of the human use antibiotics in the USA from 2010 to 2011. So the penicillins were found in the effluent of the wastewater treatment plant with with a concentration up to 1,600 nanogram per liter, and could be found in the human impact coastal water with concentration up to 78 nanogram per liter, and also found in the surface water with concentration of up to 48 nanogram per liter. So this is obvious, very remarkable concentration about the penicillins, because at the beginning, the first generation, or at the beginning, the design of this kind of compounds, the penicillins are easy and not stable, are easy to be hydrolyzed. In our study, we choose another techniques to treat this kind of compounds. Many studies have focused on the hydrolysis, alcoholysis, alkaline and protein, beta lactamase, and metal ions to remove the penicillins, and also some traditional oxidant like ozone, like chlorine dioxide, like potassium permanganate were used to treat the penicillins. But this kind of method mostly focus on the removal efficiency. There are seldom studies about advanced oxidation process used for the degradation of penicillins. And also the performance of the reactive articles on the penicillins degradation was still unknown. So in our study, we chose a novel technique, peroxide sulfate, for the degradation of penicillins. UV-PDS is an emerging AOPS technique with a very high radical generation, and also the PDS is very stable and easy to transportation, which has a promising application potential. So the UV-PDS could be an alternative choice of UV hydrogen dioxide. PDS could be activated by the UV light and generate sulfate radical. The sulfate radical has a very higher redox potential and more selective to some contaminants than the hydroxyl radical. In this study, we chose this technique to start our research work. Three selected compounds are penicillin G, we call it PG, and amoxicillin, AMX, and carbamazepine, CBN. All of this penicillin G is the first generation of the penicillins. So all of these 3 compounds have a very basic structure of PG, but AMX and CBN is the model compounds has a structure to resist the hydrolysis. This is the benzene and amine on the side chain. This is the photodegradation reactor. It's very simple with the UV lamps control. So we put the reactor inside. So this is the results. The results show that after the addition of the persulfate into the solution of penicillin, so the penicillin was degraded very quickly. The degradation of penicillin followed the first order kinetics. So penicillin degradation include direct photolysis and indirect photolysis. So direct photolysis mainly because of the UV light. Indirect photolysis mainly because of the sulfate radical and the hydroxyl radical. So we can express the indirect, the rate constants of the indirect hydrolysis as a function of the second order rate constants of sulfate radical with penicillins and the hydroxyl radical with the penicillins at the concentration of the sulfate radical and the concentration of the hydroxy radical. Competition kinetic method were used to determine the constant of the second order rate constant. From these results, we can show that the rate constants of sulfate radical to penicillin is at the same level as the hydroxyl to penicillin, but sulfate radical has a little higher rate constants. So in the next step, we want to determine the concentration of sulfate radical and the hydroxyl radical. So simplified pseudo steady state model was developed to calculate the concentration of sulfate radical and hydroxyl radical. So this model, with this model, we can modeling the degradation of the penicillins about the UV-PDS. From these results, we can see that the model could the model predicts, agree with the experimental data very well. And also we can know that sulfate with the addition of the PDS doses, so the indirect photolysis increase very significantly. That means the sulfate radical are the main contributor to the destruction of the penicillins. So we also investigated the effect of pH or penicillins degradation. Experiments was and pH 3 to pH 9. So we can see that acidic condition promote penicillin's degradation because acidic catalyst of PDS to generate sulfate radicals. And the model can predict predicts the experiments very well. And also sulfate radical is a major contributor to the penicillin's degradation and all pHs. So next, we want to know water metrics such as bicarbonates and chloride and no natural organic matters. What are their effects to the degradation of the penicillins and the UV-PDS reaction conditions? So because of the, we can see from the results that the bicarbonate and the chloride have inconstant effect on the penicillin's degradation, and the natural organic matters could inhibit the degradation of the penicillin's degradation, because the nature organic matters could absorb the UV lights and could react with the radicals. So they are a big problem when we use the UVPDS for penicillin's degradation in the real water. And also the degradation pathways were investigated. So there are two types of oxidation products, TPS-16 and TPS-32. So PPS-16, two products is occurred here with oxygen attached to the sulfur atom. And the other two products means that the active site is on the side chain of the benzene and the amine. So for the TPS-32 also there's a PG sulfone, and the other reactor is at benzene group. So from the analysis of above, we know that sulfate radical might attack all three sites of PG. Sulfur atom, benzene, and the secondary amine on the side chain. So AMX also was investigated. So AMX have primary amine on the side chain, so which the different of the structures from the PG. And from this study, we know that the reactive site is also the sulfur atom, the benzene, and the primary amine, which is similar as the PG. The degradation of penicillin in real water was investigated. So from this study, we know that the rate constants of UV of the penicillin's degradation and ultra pure water is greater than that in the surface water and greater than that in the wastewater. Because and also the penicillin's degradation in surface water and wastewater were underestimated by the models, because we didn't include the derived radicals and other conditions in this model. It's so to summarize, pseudo-state kinetic model revealed that direct and indirect photolysis both contribute to the penicillin's degradation. And the sulfate radical was the major contributor to penicillin's indirect photolysis. Bicarbonate and chloride could react with sulfate radical and hydroxy radical and generate derived radicals. So these derived radicals could have reactivity with amoxicillin. And the carbonate radical has similar reactivity with the selected penicillins. The product analysis indicated that the sulfur atom and the benzene of all the primary amine on the side chain were the reactive sites for sulfate radical oxidation. So that's the second part of my talk. And the third part actually is other techniques. I don't want to mention a lot because it's totally another story, but I do want to let you know some information about these techniques. So basically, we think about this technique is for groundwater pollution treatment. So it's a dichlorphenic treatment by the sulfide modified nanoscale zero-valent ion. We know that for the groundwater remediation, pollution remediation, usually the nanoscale zero-valent ion will be used on site. But the nanoscale zero-valent ion will not stable and easy to aggregated and settled. So we use the sulfide to modify the nanoscale zero-valent ion and to change the electron transfer direction. So that will inhibit the aggregation and the sedimentation of this material, and will accelerate the hydroxyl production, which will have a good performance on the oxidation of the DFC. So that's our target compounds. And the last technique is molecularly imprinted polymer. We call it MIP. Actually, this is not a novel technique. This technique was invented in about 1970s. But we think about this technique, with this method, we want to use it to grab the micropollutants in the environment. So we have an idea. There's a PRB underground for the treatment of underground water. So if we can make this material, we can put this material into the PRB, and then when the effluent pass through the PRB, will be the micropollutants will be absorbed or grabbed by the MIP. So that is the idea about these techniques. We published this in Applied Catalyst B, and also there's a coming paper will be published in the EST about the sulfide modified nanoscale zero-valent ion. So that's the end. Here I would like to thank you, my group, and also for the material synthesis, I would like to thank you, the UIUC, because I used to work here for about 1 year. And also I would like you to visit Shanghai, the economically developed area, and also has a very big challenge for the PPS contamination. So we can find the problems and the solutions to work together. That's all. Thank you.
We have time for one or two questions, if anyone has questions.
Yeah, thanks for the talk. It's curious how China is approaching the regulation for requiring treatment for these kind of compounds. Is the efforts just on the research end, or is there some movement to actually require treatment, or a plan somewhere down the road to require treatment?
Thank you for your question. I think until now there is no requirement for the regulation of the PPCPS in China. So yesterday I think there's a good topic about Sun from the University of North Carolina. Yeah, he mentioned that the EPA has a advisory regulation about the micro compounds control. So, but until now in China, we seldom have that kind of regulations or advisory regulations. So that's we only do research work. We are prepared for this regulation.